Hello everyone, my name is Manuel Garber. I'm a professor here at UMass. My lab works on what some people call immunogenomics, trying to understand from the basics of how cis-regulatory DNA elements establish the transcriptional response to a variety of immune stimuli, to understanding how variants affect those transcriptional responses and ultimately how they affect autoimmunity and the ability to respond to infection. And as you heard yesterday from John, I also work on more clinical relevant data using human samples to understand how transcriptional and gene regulatory networks are affected in autoimmunity and how they establish and are responsible for disease. I also run the bioinformatics core that hopefully you will find very useful, as it provides tools and a framework for you to carry out analysis of a wide range of different high throughput experiments. In ATMAS yesterday, the one that precedes this one, ATMA covers key concepts on experimental design. What are the pitfalls that folks have fall into when they analyze an experimental data that was acquired with certain biases, how to design an experiment so that you can measure something that can be compared across the conditions that you're interested in, things that may affect the usefulness of your data and might prevent or have to be corrected in order to leverage the experimental data that you encounter. In this lecture, we are going to step back a little bit, and instead of having a high-level view of what we're going to do, we're going to try to lay the basis on how mathematically we express the concepts that Athma introduced in her lecture. In particular, we're going to focus on basic probability with an aim at give you a little bit of what problems can be solved by probability thinking, and also the tools that statistics use that are probabilistic in nature. So what is the difference between probability and statistics? That's an interesting concept because they're really intertwined. But in general terms, we can say that probability is concerned with prediction. So given a model, what can we say about the future? And we call this model is the basis of probability, and we do probabilistic analysis based on this model to try to evaluate a given outcome or future outcomes. Statistics is a little bit the reverse. It is concerned from you have data. Let's try to see how can we generalize from this data. So again, in probability, we build a model, a statistical model that incorporates a probability distribution on events that we think captures how a phenomena occurs. And by assuming this model, we try to predict and estimate specifics, and sometimes to verify or identify flaws on this model. Statistics, on the other hand, provides a probabilistic framework, really, to test whether an observation is consistent with the model. That is the type of thinking we do when we have a statistic. So we have a null model and we try to see if the data that we acquired is consistent or not consistent with that model. So again, in general, we try to infer the truth or the general phenomena from a small sample, that is statistics. Instead, probability is given a model. How can we use it to understand better the future or present events that have happened and can be explained by that model? So to simplify what I just said, probability goes from general to specific. From model, we sometimes generate actual data by sampling from our model. Statistics does exactly the opposite. From a specific data set, you want to infer general principles, and from a sample, you want to infer what the population is. So typical problems that we are going to think about as probabilistic problems are, we have built a model a probability distribution on disease tests, and we want to really understand what is the probability that you're actually sick if you test positive in the test. What is the probability that if a gene knockdown affects 3 genes in a specific pathway, that this knockout gene is critical in that pathway? Or we have a probabilistic evolutionary model and want to use it to decide whether 2 sequences between human and mouse are actually orthologous to each other. We can use it. You can use the mouse sequence to study a human condition, for example. That are typical probabilistic problems. And specific things that I would hope you guys get from this lecture are basic concepts to, as I just said, estimating the exact probability or the estimated probability of having a disease given that you test positive in a test. We're also going to try to discuss enrichment analysis, right? That's the example I just gave by a knockout gene, how unlikely it is that a gene knockdown affects 3 genes in a pathway that has 1,000 genes. And I want, at the end of the lecture, to get into a more general approach called maximum likelihood that is widely used, even if you don't know that you're using it, an approach to estimate model parameters from data. So this is a highly used approach in statistics that I want to briefly discuss. And I will introduce very simple concepts, very simple distributions, and again we'll build on AFMA's lectures to discuss what a p-value is. And I also would like, as a objective of the lecture, for the few of you or the many of you that actually are interested or may not yet know that they're interested in a different approach to biology, which is a quantitative approach to biology. Maybe they find these and some of the lectures in this bootcamp exciting and may consider working in this field that we're going to just lay the basis for. The 3 main concepts that I want to lay down before we continue, and that are the basics of probability, is the outcome of all the possible outcomes that we are interested in and that are usually associated with a number, the probability or frequency at which these outcomes occur, and then events that might be a single outcome or a set of outcomes of interest. So for example, an outcome of developing or not developing cancer can be used to build many different events, like a number of mice develop cancer in a number of treated individuals, or that it fell 10.3 inches of rain in August 10th. Those are typical events. Probabilities are defined on outcomes, and events that are built of these outcomes can be computed their probability as well. So if we define the probability of rain and the probability of no rain, then we can compute probability of more complex events, like what's the probability that it will rain in the next 3 days, or it will rain in one of the next 3. For probability distributions that we can enumerate all the possible events, we can just associate every single event with the probability or frequency that event occurs. For probability distributions that are continuous, that take on any possible number or a subset of all the real numbers, then the probability of any single event is of course 0. So for probability distributions in which you can enumerate all the possible outcomes, then you can just enumerate the probability or frequency that each event occurs. But most of the probability distributions that we really think about are not discrete, are continuous. For those, instead of the probability of each event, what we use is the density distribution. And that density distribution allows you to compute the probability that an event is within an interval. So for example, if we are looking at the distribution of fat in a given type of butter, then the probability that, say, the Guernsey butter that you bought has between 5 and 6% of fat is the shaded region that I'm showing, which is the area under this density function for the Guernsey amount of fat per butter, per block of butter that you buy, and that is that area under that curve is the probability that yes, the block of butter that you bought has a fat between 5 to 6% of fat. So how do we compute probability of complex events? Let's start with the simple case where you have events that do not share outcomes. They are mutually exclusive. So for example, it can rain or it cannot rain, but you cannot have both at the same time. Or if you have two alleles, any biallelic genotype can just be one of the four possible combinations. So those are four events, and they are independent of each other. Another way to visualize events is by displaying different events as the individual outcomes that they represent, and whenever they have common outcomes, then they overlap. So for example, the A and B3 are independent, they don't share any outcome, they are disjoint. But B1, B2, and B3 are all independent, they don't share any common outcome. But there are interesting things when they do share outcomes, and that makes computing the probability of them both or both combined complicated. For example, if we know that event B1 or B2 occurred, we know a lot about what happens or what's the likelihood that event A occurs. So when you have two disjoint events, for example, computing the probability that either one of them occurs is simple because it's the union of all the possible outcomes or the sum of the probability of each of the events separately. So if you want to compute the probability that an individual has a blood type of O, then you can sum the probability that an individual has a blood type of O negative plus the probability that an individual has a blood type of O minus. So this helps tremendously to compute more complicated probabilities. So for example, if we want to compute the probability that a child of two heterozygous parents inherits a dominant allele, then there are only four independent outcomes, and they all have the same probability. So the probability of inheriting one is the sum of the probabilities of the three events shaded in yellow, which is 3/4. So to summarize, when an occurrence of one event does not inform of the occurrence of another, they don't share any common outcomes, they are considered independent events. To compute the probability of two independent events occurring, you just multiply the probability of both events. Just to show one more example, if we have two heterozygous parents but now on two alleles rather than just one, what is the probability that a child is also heterozygous on both alleles? So we could solve this by writing down each and every single of the events. All of them have the same probability, and then just summing up the events of interest and computing the probability of all those four events circled in red, which is 1/4 of all the possible events. So the probability of being heterozygous is 1/4. You can see how the product rule comes handy, because we can compute the same probability by computing independently the probability of being heterozygous for the B allele and the probability of being heterozygous for the C allele, and then the product rule, because we are assuming that these are alleles that are not linked, so inherit them as they are independent of each other. So the probability of being heterozygous is the product of the independent probabilities of being heterozygous in each of the two alleles. And you can see that now we can generalize not just to two alleles but to many independent alleles, and the probability of being heterozygous in 12 non-linked alleles, the product of being heterozygous in each one, the product of all the probabilities of being heterozygous in each one, making this computation much simpler. Now, if you know that B1 occurred, then you know a lot about what happened with A, right? Similarly, if you know that B3 occurred, you know that, for example, A cannot have occurred because B3 and A share no events, so they are independent. But if B2 or B1 occurred, as I mentioned at the beginning, then you know a lot about A. So those probabilities of one event, given that you know that a second one occurred, are called conditional probabilities, and they are non-zero whenever the events are not independent, they are dependent on each other. So let's think a little bit about this conditionality. What happens with A if you know that one event that is on which A is dependent occurred? So what is the probability that A happens if you know that B2 happens? So A happens only when an outcome within the intersection of A and B2 occurs. A does not happen whenever the outcome comes from the complement of B2 and A. So you can only draw, or the frequency by which A occurs is the frequency by which you can sample from the intersection with respect to all the possible outcomes in B2, which is shown here as 3/4. 3/4 of the time, an event from B2 overlaps A, and hence 3/4 of the time, if you know that B2 happens, you have A also happening. We can play this game with any of the events that's shown here. If B1 occurs, A has to occur because B1 is contained in A. As we just discussed, the probability that A occurs if you know that B2 occurred is just 3/4. 3/4 of the events in B2 are shared with A. B3 and A are independent, and hence the probability that A occurs if you know that B3 occurs is 0. And you should think about these probabilities that are the opposite, right? What's the probability that each B1, B2, and B3 occurred if you know that A occurred? And conditional probabilities are very common, and actually it's the topic in which we're going to spend a lot of time today. So for example, the probability that it rains, given that it's clouded, is much higher than the probability that it rains if we don't know what the sky looks like. So, and I want to stress that condition is not commutative. Conditionality is not commutative, right? So the probability that rains, if you know it's cloudy, it is not the same as the probability that is cloudy if it's raining, which is almost 1. It seldom rains when you can see the sun on the top. And a probability that we're going to devote a lot of time today is, for example, what happens if you test positive in our weekly COVID test? What is the probability you actually are infected by COVID, given that the qPCR result is positive? And again, I just want to stress that the probability that you tested positive, if you know that you have COVID, is a different one, and this is called the sensitivity of the test, right? What is the fraction of times that the test captures or results in a positive when you are infected? That is different than the more interesting probability or more relevant probability, but for our discussion today, which is you tested positive. Are you infected? Do you have COVID? So that is what we're going to discuss for a while. So what it's really incredible, right? And although the theorem itself is trivial, what it does is really amazing. That these two probabilities that we're discussing, the probability of having an infection when you test positive for a test, and the probability of testing positive if you have the disease, are really intertwined. And this intertwining is what people call the Bayes theorem. And I want to derive this theorem for you guys because I think it helps your intuition. And the Bayes theorem relies on the fact that when two events are not independent, then the joint probability that they both happen at the same time, as we discussed before in the product rule of two independent events is not the product of the independent probabilities. What is this probability of a joint? What is the probability of A and B occurring if you know, or if A and B are dependent, that conditional probability? It is intuitive. We discussed it before. The probability that both A and B occurred is, you can write it in two ways. It's a probability that A occurs if B occurs times the probability that B occurred, or it is the probability of B occurring given that A had occurred plus the probability that A occurs. So because you can write these both ways, then they are intertwined. And we are going to use it right now to discuss this problem that I posed a few slides back, which is, what is the probability of having COVID if you test positive? So what's the probability of both simultaneously having COVID and testing positive for COVID? Well, you can write it as the probability of having COVID given that you tested positive for the test times the background, the regular probability of testing positive, anybody testing positive. Or you can write it as the probability of a positive test if you are actually infected with COVID times the background rate at which people are infected by COVID. So the two ways in which we wrote this joint probability are the same, so we can equate both sides of this equation. And what we're interested in is in this posterior probability, right? So now that you know that you tested positive, how likely it is that you actually have the disease. And we solve for it, so we are interested in that conditional probability. And this now can be written as three factors that we can compute. And in general, the Bayes theorem can be written like that. I just was using it for our specific example. And the two conditional probabilities in this formula come very often in statistical probability and have different interpretations depending on the problem. So you tested positive on your weekly COVID test. From my reading of the literature, and this can vary, actually, I hope in your workshop you're going to see what the relationship between all these numbers are. I found that the specificity, the accuracy of the qPCR for COVID is around 98-99%. That means that 98% of the time that the qPCR is positive, the person who was tested had COVID. But it's interesting, right? The qPCR for COVID is not that sensitive because of many reasons. Actually, I encourage you to read that the time in which you take it is different, the level at which people are infected, the way in which people take the test. All of these impact the sensitivity of the test, which is roughly, as far as I could tell, as I could find, 80%. So if you test positive for the qPCR next week, really, what is the chance that you actually have COVID? And let's write it as this condition probability that we've been discussing. So we want the probability of having COVID given that your qPCR test is positive. We write this using the Bayes theorem as the probability of testing positive in the qPCR test given that you have COVID times the background probability or the frequency or the rate of infection in our community. And the denominator, this is over the background probability or all the possibilities in which the test can actually be positive. So let's go one by one. What's the probability, the first conditional probability? What's the probability that your COVID test is positive if you have COVID? That is exactly what we discussed before as a sensitivity, that's 0.8. The probability of having COVID in our community, and I might be wrong, so I recall in my conversations with my colleagues that it's about 0.1%. So that's the rate in our community. And again, it might change, and we should watch the news to see if this changes, and I encourage you to play with this background probability. And the probability of actually testing positive can be decomposed in two independent events. testing positive if you have COVID, and testing positive if you do not have COVID. And the first one is the sensitivity, as we discussed, times the background infection rate, 0.01. And the second one is the one, or it's the false positive rate, 1 minus specificity, times the frequency at which people do not have COVID, right? 1 minus 0.01, which is written here. So the sum of both is the probability of your qPCR test being positive. So it is amazing, right? Because if we plug this in, if you test positive next week for the first time, of course, the probability you actually have COVID is just 5%. And why is that? Let's now look at it a little bit. Yes, so why is this? So look, the infection rate, as far as I knew, right, it's so low that most of the time that you test somebody, he or she does not have COVID. So think about it. If your specificity is 98% and you test 1,000 people, 20 of them that do not have COVID, will actually test positive. And at 1%, at 0.1% infection rate, you just expect one and only one of them to have COVID. So that's why most of the people who actually test positive do not have COVID. I also want to explain why we call this a posterior probability, right? So if we think of the probability or the rate of infection, 0.1%, as our prior probability, right? As our prior belief that you, chosen randomly from our population, has COVID, then the Bayes theorem tells you how much that belief or that confidence that the person that you chose by chance has COVID changes if you know that this person has now tested positive for their COVID test. So in general, we call that the posterior probability of having COVID now that you learn more about this event, in this case that you tested positive for COVID. And yes, this prior, the frequency at which, or the frequency that our population has COVID, has a huge impact on this result, as we will see in a second. So I did a little bit of playing with the infection rate. So I varied the infection rate from 0.01 as I did the calculations above to 1%. And look how much the posterior probability changes. It goes from the one that we computed 0.5 to 35% chance of having COVID if the rate of infection is 1%. And this graph also depends not just on the rate of infection, but of the specificity of the test. And look how much the posterior probability changes if the specificity just changes by 0.01. So if the specificity goes from 0.985 to 0.995, look how much this curve changes. Now, the posterior probability of having COVID, if you test positive and the infection or the background infection rate is 1% and the specificity of it is 9.995, is almost 70%, right? So it almost duplicated just by changing the sensitivity rate. That's how these calculations are dependent on the assumptions that we made in our models. So let's look also how the posterior probability changes as the other parameters change. So I just showed you two differences, how strong a difference the sensitivity makes on our calculation. If I just vary it from 98% all the way to 100%, look how much the posterior probability changes. And actually there is a huge change after 99.5 probability. And it's interesting because the FDA approved test for HIV, the Oral Quick Saliva test, actually has a 999 sensitivity rate. And look how it changes, how the posterior probability of having HIV changes at the same background as we are working on for the COVID infection rate if the sensitivity is that high, right? The posterior probability that shoots up, and that's why we want tests to be extremely sensitive, if not so specific as the plot in the right shows. If you change the sensitivity, you really don't get that much bang for the buck. The bang for the buck is in the specificity of the test. You want the test to be extremely specific for having a high confidence that the person that you tested actually has the disease. So both the background infection rate and the specificity of the test are critical in interpreting or in how much alarmed you should be if you test positive for a test like the qPCR COVID test. I want to finish this section by pointing out the way in which we computed these numbers and the probability that your test is positive. And the key was breaking it out into two independent, or into finding an independent event that we can use to condition the event in question so that it is easier to compute. And that is the general way in which that denominator in the Bayes theorem is computed. And it's called the law of total probability, that enables us to find the probability of one event by breaking it up into mutually exclusive conditioned sub events that allow you to then sum up the probability of this more complicated event into the independent probabilities that are conditioned on the ones that are more easier computed. So let's look now at another conditional probability, perhaps a little bit more complicated. We call it the likelihood of a model. So how well a model fits or explains the data observed. The likelihood is then the conditional probability of the observed data given the model. And in general, we let model parameters, say the mean and variance of a distribution, vary, and see how the different parameters explain the data observed by computing this conditional probability. So in general, one intuitive way to find the best or an interesting set of parameters for a model is to find those that maximize this probability, right? So that will be, for that given probability distribution, the best parameters that fit the data that we have. And this is something that you do every day for those of you who like statistics, because the maximum likelihood estimator of the mean of a distribution is, yes, as you would have guessed, the sample mean. And I will, just for completion, will post the proof of this fact using calculus. And maximum likelihood is something that I want to discuss because you are going to see it over and over. It is a very intuitive, although at first sight may be a little bit complicated, but it's used throughout most of the tools that you're actually going to use in this bootcamp and probably through your career. And in this sheet that you will use next week to estimate differential expression, the maximum likelihood is used to estimate the dispersion of the genes in a given expression range. Our SAM, which actually is what we use to compute the expression levels of genes from our RNA sequencing experiment, uses maximum likelihood to estimate or to figure out where reads that might be mapped to multiple genes actually should have come from. So they compute the probability that a given read comes from a transcript or an isoform of a gene. I have used maximum likelihood to estimate the pattern of substitution from a multiple sequence alignment of many different species to estimate or to understand how a given nucleotide evolves throughout the mammalian lineage. And there are many similar examples. Many tools that you use rely on this simple concept to find the parameters that maximize the probability of the data that you have gathered to estimate, to get the parameters of a distribution that we think explains or is good fit for the data. The log likelihood also can be used as a testing tool, and it's the most probabilistic way to deal with testing. So yes, this is where statistics and probability are meshed up, And although I am going to discuss statistical testing next week, this is a really nice example of a probabilistic way of thinking about tests. So let's go back to an example that is simple yet not very relevant, which is dice. And when you roll a dice, your null assumption, your naive assumption is that all sides of the dice are equally likely. So the likelihood of this model is whatever number of sixes that you have rolled among n tries that you have, where the probability of drawing a 6 is exactly 1/6. So how do we compute this? I want to take this to also introduce a very useful distribution, the binomial distribution, which is the probability of k successes in n tries, and is a distribution that will come over and over. It's one of the most useful distributions that there are, and it depends on this number, because when you draw n samples from a distribution, or when you have n events, you can draw k successes in many different ways, right? You can have the first k being a success, and then the last n minus k being success, and you can have the first being a success, then no successes, and then at the end you have successes. So all these ways are equally likely, and the way to compute this is by this beautiful binomial coefficient that we say in how many ways you can choose k in n tries. And the formula for that is here. I will not derive it. You can use Khan Academy for a beautiful description of the binomial coefficient. But once you have that number, then the probability of observing k successes on n tries is the number of ways in which you can do this, that's the binomial coefficient, time the probability of each one, the probability of having k successes, and followed, say, by n minus k failures is the probability of each success, p k times, that's p to the k, times the complementary probability, 1 minus p, n minus k times, that's 1 minus p raised to the n minus k. So with this sets up a good framework to discuss whether a dice is loaded or not loaded, given that you observe some number of sixes in a number of trials. So we're going to compare two models, right? Model 1, all faces have equal probability. The probability of a 6 is then 1/6. And the model 2, maybe 6 has a different probability. And we're going to use maximum likelihood to estimate the probability of drawing a 6 based on the data that we have. And we will compare those two models using the ratio of their likelihood. And that is an example that is very simple, but yet representative of the more complicated cases that I showed you before. So let's do this for a toy example, for a fun example. So say we rolled, or somebody in the casino rolled 58 sixes in 500 rolls. So we've been counting how many times the sixes come up. So remember that our model 1 was an equal probability for all faces, so we can compute the likelihood of these 58 sixes in 500 rolls by the binomial distribution. So I'm just plugging in the numbers from the formula we had before, that is all the ways in which you can roll 58 sixes when you roll 500 times, that's the first coefficient, followed by the probability of drawing 58 sixes and the probability of not drawing sixes in the remaining times. And again, M1 is the model where the probability of 6 is just 1/6. These numbers are super tiny. They are minuscule. So we usually do not use the likelihood. We always work with the logarithm of the likelihood. And in fact, just taking the logarithm of that product, for those of you who do not remember, the log of the product is the sum of the logs. So also logs, the logarithm is a great transformation. We'll use it over and over, even in this bootcamp, as you will see going forward, because it makes products into sums. And if you remember when you were in primary school, you probably hated to take products, but you didn't mind summing up. And again, in math, sums are always easier to deal with than products. So the fact that the logarithm changes products into sums, super useful. And it is also beautiful because if the original function was, say, increasing, the log of that function keeps that property. So the maximum of the point in which an original function achieves its maximum is the same as the point in which the logarithm of that function achieves the maximum. So the logarithm has these beautiful properties, and that's why you will see a log transformation done for many, many different things. But anyway, I digressed. The logarithmic of that likelihood of the model 1, if you just plug in the numbers and compute it, is around -8. Again, it's a tiny number because it's the original likelihood is e to the -8, right? So yes, drawing 58 times is a little bit low. We can test our null hypothesis to see if all the dices are equally likely by comparing the likelihood of this model to the likelihood of the model that we would obtain by fitting or by figuring out the maximum likelihood estimator for the probability of drawing a 6. And the maximum likelihood estimator for P, the probability of success for a binomial distribution, you are not going to be surprised, is the proportion of successes, right? So in this case, this estimator, and we usually use this hat to note or to denote the estimator of our parameter of a model, is 58 over 500, which is, around 1.116. And again, as I will do for the normal distribution, I will post the calculus derivation of why the maximum likelihood estimator of the probability of success of binomial distribution is the frequency or the proportion of successes. For those of you who are interested, I do not think most of you will even look at it, but I feel that this just complements what I'm doing here. So our second model, the one we want to compare just to test our hypothesis, is a model where the probability of 6 is this maximum likelihood estimator, 0.116, and its likelihood is, again, just by plugging out the numbers, minus 2.9. You can see that this likelihood is 2.5 times greater than the likelihood of model first. And in fact, I will not go over this because it will require more math than we do in this course. There is a beautiful theorem that says that when you have enough samples, the ratio of the log likelihoods of two models actually roughly distributes chi-squared. This is a distribution that we are going to discuss the next day. And chi-square is one of the most common distributions used in statistics, and there are tables for that, so you can look at the ratio that we compute. This is a chi-square with 1 degree of freedom, so you can compute the significance of this ratio to around 2.5. And the probability of observing such value or greater ratio is just about 10%. So under the null hypothesis, a value of this or greater is rare. So you might think there is enough evidence to think that the probability of it drawing a 6 is not the same as the probability of drawing all the other dice.